So today's video is going to focus on the actual formation of the urine and the production of the urine. So the urine is formed in 3 steps: filtration, reabsorption, and secretion. And we're going to look at each step individually, starting with filtration. So filtration is going to be the step where water, small dissolved molecules, they're going to diffuse into the glomerular capsule. So if you look at this kind of simplified view down here, you can see the filtration happening here at step 1, where you have the glomerulus here with those glomerular capillaries inside the capsule, and the water and small molecules are going to diffuse through those filtration sites that are formed where the podocytes wrap around the capillaries. Remember, the podocytes were those cells that had like those feet-like appendages, and they wrapped around the capillaries, forming those filtration slits. Okay, so that fluid is going to come out of the capillaries and into this glomerular capsule. At this point in time, the body is not very picky about what is going through right now. It's not filtering good from bad right now. It's filtering just in general substances from the blood. And that glomerular filtrate is that substance that is left over or going into the capsule, right? So that's the water that you'll find there in the capsule. Okay, some of the things that can cross that filtration membrane are water, amino acids, urea, sodium, glucose, all of those things are small enough to cross, right? So these can end up in the filtrate. The larger things like blood cells and sorry, I was looking at my tea here. It looks funny. So the larger cells, like blood cells, some of the proteins that are in the blood, like for example, albumin, these are too large. So those will not cross that filtration membrane. That filtration membrane is going to be made of those podocytes, right? So those cells that wrap around the capillaries there. So it's going to be made of those podocytes, the endothelial cells in the capillaries. So essentially the cells that make up the cell wall of the capillary. So those endothelial cells of the capillaries, as well as the basement membrane of the capillary cell wall. Or not cell wall, that's probably a bad term for it, of that capillary endothelial membrane. So that's going to make up what we call our filtration membrane. So the small molecules, they're going to go through that filtration membrane and they're going to end up in this filtrate that's in the capsule. So let's take a look at how this filtration process occurs. So there's essentially 2 pressures that are going to help move fluids back and forth, fluids and molecules back and forth across the various cells and across these various membranes. So the first one here is hydrostatic pressure. Hydro is water, right? So hydrostatic pressure is going to be pushing water from a high pressure to a low pressure. So not talking about concentration or anything. We're talking about actual pressure here. So hydrostatic pressure is pushing water from a high pressure to a low pressure. You can see on this image over here, we've got some hydrostatic pressure pushing water out of the glomerular capillaries and a small amount of hydrostatic pressure pushing some filtrate towards the capillaries. The other one is osmotic pressure. Osmotic pressure is created by the dissolved substances that are in the fluid. So those salts, those various ions like sodium, potassium, those are going to help cause the osmotic pressure. This will pull the water to areas with a higher solute concentration. So if we keep in mind some of our 9th grade biology here, right? Solute, that's the dissolved substance that's going to be in the fluid. So if the solute concentration goes up, then the water concentration has to go down if the solute concentration goes up. So essentially what this is doing is attracting water to areas that have a lot of dissolved substances in them. Water is always going to flow from a high to a low concentration. So if we look at this image over here, all right, we have this osmotic pressure here saying that it's going into the glomerular capillaries. So what that means is that there is a higher water out in the capsule, lower water inside the capillaries because of this right here, because of those proteins, those big proteins that stay in those capillaries. That lowers the water concentration of the capillaries, so it naturally pulls water in towards the capillaries. So you'll notice these two pressures go in opposite directions, but there is still a net push of fluids and solutes and such out of the capillaries and into the capsule. So this is all part of filtration and actually forming that filtrate solution that's now going to move into the tubule. So now we get to step 2 in urine formation, which is the reabsorption step. So we have this filtrate that was made right here, right in the glomerulus with the fluids and the substances leaving the glomerulus going into the capsule. So now, because we said when that filtrate was being made, the body wasn't being very picky, right? So now we need to reabsorb all the good stuff so we can get rid of the bad and the extra stuff. And so this is going to start that process. So the filtrate has moved from the glomerular capsule into the renal tubule. If you remember, the renal tubule has 3 basic parts, right? The proximal convoluted tubule, the nephron loop, and then the distal convoluted tubule. So most of this reabsorption is going to occur in the proximal convoluted tubule. So the water, the dissolved substances, they're going to be reabsorbed into the capillaries that are going to surround the tubule. If you think from back to your pictures on the previous video, you'll notice there was lots of capillaries that surrounded these, not just one little loop. So that's where the water and substances are being reabsorbed back into those capillaries that are surrounding the tubule. Most of this reabsorption is going to happen in the proximal convoluted tubule. Some will happen in the distal convoluted tubule and some in the collecting duct. So you notice we didn't mention in the nephron loop portion. So again, the bulk of this reabsorption is happening in the proximal convoluted tubule. The distal convoluted tubule does a little, as well as the collecting duct will do a little bit of reabsorption. The proximal convoluted tubule has microvilli to help increase its surface area, which will help with this reabsorption. Kind of like how the intestines had the folds and then the villi, and then the microvilli to help with the absorption, increase the surface area for the absorption of food. The proximal convoluted tubule will increase its surface area so that it can reabsorb even more. If you look at your image down here on the bottom right, we've zoomed in. This is the tubule side. These are the paratubular capillaries. Remember, peri means around. So these are the paratubular capillaries, and you'll see that things are being reabsorbed back into the capillaries. Water, sodium, potassium, various other ions. So let's look at step 3 in urine formation, which is going to be the secretion. So we have filtration, reabsorption, and now we have secretion. So during secretion, what we're actually secreting is these waste products. So we're actively pumping waste products out of those capillaries that surround the tubules and into the renal tubule. The fluid that is left in the renal tubule is what is going to become the urine. So we are pumping waste products out of those capillaries that surround the tubule and directly into the tubule. So they're actively pumping them. So this is going to require energy to do this because it's a form of active transport. Those tubular epithelial cells, so again, cells in the proximal convoluted tubule, in the distal convoluted tubule, as well as in the collecting duct, these are where we're going to be actively pushing those molecules out into the lumen. Remember, the lumen is the space. So that would be the space into the space in that renal tubule. We have a picture coming up in just a minute that will help, I think, with some of this. So during this process as well, during secretion, is where our kidneys can help maintain our blood pH. Blood pH is normally maintained around about 7.4. And so the kidneys can help maintain that by secreting hydrogen ions or bicarbonate. So those tubular epithelial cells, they can secrete hydrogen ions, which would make something more acidic. So that would help then to bring the pH up if we needed to, because I'm secreting the hydrogen ions. So I'm taking acid, taking these hydrogen ions that make the blood more acidic and secreting them into the lumen so that they go out in the urine. Or they could secrete bicarbonate, which is HCO3, which is alkaline. So if the blood was too alkaline or too basic, then we could secrete some of these bicarbonate ions into the urine so that the pH would lower and become more acidic. So they're helping to maintain that blood pH of approximately 7.4. Also, some drugs can be secreted here as well out of the bloodstream and into the kidneys. Things like penicillin is filtered out by the kidneys. Aspirin is filtered out by the kidneys during this secretion process. So let's look at this this kind of summary picture quickly here. So first, again, we have step 1. We have this filtration process in the glomerulus that is bringing the filtrate out using that hydrostatic, that osmotic pressure, forcing that filtrate fluid out of the capillaries into the proximal convoluted tubule. From there, we move into the reabsorption portions, happening again mostly in the proximal convoluted tubule, a little in distal, little in the collecting duct, but reabsorbing the good things back in to those surrounding capillaries before we then move on to step 3, which is that secretion, where we can then actively pump waste products out of the capillaries and into the lumen so that it can be excreted in the urine. So you'll see there's some things that these are the things that can go back and forth during filtration as the solute plasma components. During secretion, we've got some of these ions, some drugs. During reabsorption, these are good things that we'd be putting back into the body: glucose, lactate, amino acids, just trying to maintain homeostasis by regulating this fluid. Okay, so far we've been focused more on this renal cortex, which was that outer portion of the kidneys here. That's where I'm finding that glomerulus with the PCT and the DCT. And you notice we keep leaving out the loop of Henle, or the nephron loop, right? We leave that out, and we haven't been talking about the fact that it's doing reabsorption or secretion or anything like that. So sometimes that loop is long enough where it goes all the way down into the renal medulla, where the renal medulla forms those pyramids that have those papillae at the end that will eventually empty into the renal pelvis, that kind of central part there of the kidney. So in the renal medulla, I don't have any active reabsorption or secretion happening in those descending and ascending limbs of the nephron. So the descending and ascending is just which direction the fluid is flowing. So I've got a descending limb on this side and an ascending limb going back up that loop there. So in the descending limb, some water is able to leave the filtrate and go out into the kidney. In the ascending limb, sodium can be actively reabsorbed. However, water is not able to. There are no, I don't know if you noticed on one of the previous pictures, but there was something called an aquaporin channel. Water to easily flow back and forth across cell membranes. Water needs these aquaporin, right? Pore opening needs these aquaporin channels. And so in the ascending limb, while sodium is being reabsorbed back into the capillaries, water is unable to follow it because there are none of these aquaporin channels. So we get this very concentrated, very salty interstitial fluid down in the renal medulla. Remember, interstitial fluid is that fluid that is surrounding our cells. So what we have in the renal medulla portion of the kidney is going to be very concentrated, very salty, because water was not following that reabsorption process that the sodium was doing. Okay, so we have the basics of how the urine is actually formed. So now we're going to talk a little bit about urine regulation, like urine formation regulation, and then actually getting it out of your body, and we'll be all set. So a lot of urine production is controlled by hormones. We've talked about how they do, long-term control, maintain homeostasis. They kind of are running your machine for you. So the first one we're going to talk about is aldosterone. Aldosterone is a steroid hormone, meaning it's a steroid lipid-based hormone. It is produced in the adrenal cortex. If you remember, the adrenal glands sit right there on top of the kidneys. So aldosterone is produced there in the adrenal cortex. It is released when your body detects a decrease in sodium concentrations. So when your body detects a decrease in sodium concentrations or a rise in potassium concentrations. So your body is monitoring your blood levels of sodium, potassium. These are really important for nerve function, for membrane channels. And so we've got to maintain certain levels of sodium and potassium. So when our sodium drops too low or our potassium gets too high, aldosterone is secreted by the adrenal glands. Another way that aldosterone could be secreted by the adrenal glands is by a drop in blood pressure. So a drop in blood pressure can stimulate aldosterone because a drop in blood pressure increases renin secretion. If you remember this from our cardiovascular unit, right? Renin turns angiotensinogen into angiotensin, just like our pepsinogen was converted into active pepsin. So it turns angiotensinogen into angiotensin. When that angiotensin kicks in, that's going to constrict the blood vessels, which will help increase blood pressure. And during that conversion of angiotensinogen to angiotensin, that's going to stimulate the adrenal cells to secrete aldosterone. Aldosterone, again, remember, was also secreted when our ion balances got off. Sodium got too low and potassium got too high. So what aldosterone will do is it will increase sodium reabsorption and potassium secretion. So it's going to act on the distal convoluted tubule and the collecting duct. And when it increases the sodium reabsorption, water will follow that sodium. So water will follow the sodium. So water will be reabsorbed with the sodium back into the capillaries. So what that will do is that will decrease the amount of water that's in the urine. So the urine itself will have lower sodium levels, lower water levels, but higher potassium levels. And so what that will do is that will retain more water for us to stay in the blood circulation, which will in turn increase blood pressure. So when that blood pressure drops, it's to your body's benefit, right, to try to retain water, because that's another way besides constricting the blood vessels that we could help increase blood pressure. So that's what that aldosterone can help us with. Another hormone that can help regulate urine formation is atrial natriuretic peptide, or ANP. This is going to be made within the actual atria of the heart. Remember, those were those receiving chambers, those superior chambers that were receiving blood from the systemic circulation and from the pulmonary circulation. So this ANP, or atrial natriuretic peptide, is released when the atria are overstretched. So if they are overstretched, what that is telling us is that our blood volume is going to be very high. So the blood volume is too high, and so that's causing that means the atria are essentially receiving too much blood, and it's overstretching the walls of the atria. So what this hormone will do is it will actually inhibit sodium reabsorption in those collecting ducts. And the opposite of aldosterone, if I'm not reabsorbing the sodium, then again, the water will stay with the sodium. So I get a decrease in water reabsorption as well, which will then in turn, what that will do to the urine is that will increase the volume of the urine. That will increase the sodium within the urine. So in my blood now, I have decreased, So now within the blood, I have decreased the blood volume because I've excreted more water in the urine. Excreted more sodium, excreted more water in the urine. So atrial natriuretic peptide and the aldosterone essentially do almost the exact opposite of one another. Okay, our third and final hormone that we're going to look at is antidiuretic hormone. Anti means against or not. Diuresis is the fancy term for one of the fancy terms for urination, losing water. So antidiuretic hormone is going to be against that. This is also called vasopressin. So you could see either one, ADH or vasopressin. This is secreted when blood osmolality increases. Remember, osmolality is the solutes that are in the blood. So if I increase my solutes, my dissolved substances in the blood, then that makes my water concentration go down. So ADH is going to be secreted when we are getting dehydrated. And so we will secrete this ADH to try to concentrate the urine and retain some water. So the blood osmolality will increase when we are dehydrated, which makes sense, right? My water concentration drops, then that would make my solute concentration mean it's going up. So the ADH causes the collecting duct cells to produce more aquaporin molecules. So if they produce more aquaporin molecules and insert them in the membrane, then that increases their water reabsorption. So remember, those aquaporins are those channels that allow water to go back and forth across the membranes easily. So in the collecting duct, if I'm putting in more of these channels, then I'm able to reabsorb more sodium or more water from the collecting ducts, which would mean it would be retained. And so I would have a very concentrated urine, but I would be trying to retain water some to essentially rehydrate the body. When the body is well hydrated, it produces less ADH, and we end up with this very dilute urine because we're not trying to, our blood osmolality level is within normal ranges, so we're not trying to make sure that blood osmolality isn't getting too high. Okay, so we've made urine, we have controlled when it's getting made, why it's getting made. So let's look at how it's stored and removed from the body. So the remaining portions of the urinary system, their job is all to either move or excrete urine, not to make it. The kidneys are the only organs that are actually making it. So the ureters are these tubes right here. So the ureters are going to run from the kidneys, in particular the renal pelvis of the kidneys down to the urinary bladder. And they're going to be lined with smooth muscle. Oops, I don't know, I put two N's in that. They're going to be lined with smooth muscle that is going to do peristalsis, just like the digestive system. So there will be peristalsis movement of this smooth muscle to help push the urine down from the renal pelvis of the kidneys down to the bladder where it will be stored. So the bladder is this hollow organ down here. It is a hollow organ lined with smooth muscle so that we can remove the urine from the body. It's going to release the urine from the body through this small, it's different sizes in male and females, but this shorter tube here called the urethra. So the urethra is what's going to actually connect the bladder to the outside. So it will run from the bladder to the outside of the body. In males, it's approximately 8 inches long because it travels through the penis in males. In female, it's only about 1.5 inches long because it is traveling separate from the vagina, but doesn't have to go through a penis, so it's not quite as long. Okay, so finally, we're going to excrete the urine from the body. So when we excrete the urine from the body, like we've already talked about, we have peristalsis, moving the urine forward down the ureters, down to the bladder. And then we have what's called micturition, which is the actual process of urination, or it's also sometimes called voiding. You void the bladder. So that's the process. That's the actual process of urination happening. So when this process happens, the bladder will contract, because remember it's a muscular organ. So it will contract to forcibly push urine out. The opening of the bladder is controlled by 2 sphincter muscles. Remember, sphincter muscles are round muscles that control things opening and closing. So we have the internal urethral sphincter. This one is under involuntary control. This is the one that when our bladder is full, this sphincter muscle relaxes and it tells us, oh, our bladder's full, I need to go potty. The external urethral sphincter, this one is going to be skeletal muscle, so it will be voluntary control. And this is the sphincter muscle that controls the actual opening to the outside. This is why you can hold your urine, right? Because you have that skeletal muscle opening all the way to the outside. So when the bladder fills and stretches, that's going to send a signal to the spinal cord, in particular to the sacral region of the spinal cord. So the bladder fills with urine. That sends a nervous system signal to the sacral region of the spinal cord. That nervous system signal is going to tell the muscles to contract and relax, right? It's going to tell the actual bladder muscles that it's time to contract. It's going to tell that internal urethral sphincter muscle that it is time to relax, that it's full and we need to empty that bladder. At this same time, the brain is getting a message that the bladder is full. So that's how you know that this is happening. So you get that message when that internal sphincter relaxes. That sends that message to the brain, and now your brain can determine whether it's the right time or not for actual voiding to happen. When it is the right time for actual voiding to happen, the brain will send a message that it's time to relax that external urethral sphincter muscle and actually let the urine out. Right, so I think this is going to be it for our chapter on the urinary system. I think we've gone into enough detail for our purposes in this class. So we'll continue to try to work with this material, make sure we get it down, do some labs, okay, and maybe do some analysis of some different urine samples and such.